Presentation, we're going to be looking at the idea of DNA profiling, or DNA fingerprinting as it's sometimes called. And as that title suggests, this is a way that we can actually show a unique profile for each individual. Okay, so we inherit half our DNA from each of our parents. This is going to be relevant later. And a large proportion of our DNA is made up of long sections of highly repetitive sequences. Now, most of our DNA is virtually identical to the DNA of other people. However, some of these highly repetitive sequences within the human population do vary quite widely, and these sections are known as short tandem repeats. So if we look at this diagram of the proportion of DNA within a human, we can see that actually only this part here is actually varies widely. We do have these bits, the exons, and we've looked at these before. The exons are sort of the alleles that we've looked for that might give us different characteristics, but often these will only vary by one or two base pairs. So these bits we can't really use to analyse and look at a DNA fingerprint, but these bits vary a lot more. So these are the bits we're going to be looking at in DNA profiling. Okay, so short tandem repeats, what are they? Well, they are very, very important in DNA profiling because these are the bits that we actually look at to create this unique profile. They're the bits of our genome that are the most different from each other, and they show significant variation between individuals. So what exactly are these STRs, or short tandem repeats? Well, they contain sequences of DNA of between 2 and 5 base pairs. In fact, most commonly, they consist of 4 bases repeating many, many times. And the number of repeats varies between individuals, and these variations are known as polymorphisms. So if we look at this little diagram here, we can see that participant 1 has 1, 2, 3, 4, 5, 6, 7, 8. You can see on the top there. Whereas participant 2 has 9, and participant 3 also has 9, but participant 4 has 10. So we can see within this particular locus, this particular position in the genome, you can get these varying lengths of these sections of DNA. And that is what's going to be important later when we come to analyse them, because it's the length of those fragments that will actually give us the different profiles. Now, if we were just to look at one of these STRs, one of these short tandem repeat sections of DNA, a lot of people would have the same length, and we can see here participant 2 and participant 3 have exactly the same length. So we don't just look at one, we actually look at something like 13 different points within the genome where these short tandem repeats exist. And you have to look at all 13 and compare those to other people to get a unique profile. So for instance, if you're closely related to somebody, a lot of those short tandem repeats will be the same length. Whereas if you're distantly related, or you're not related very much at all, then you won't share many of them. So it's a way also of looking at how closely related you are to somebody. You can look at these short tandem repeats. Okay, so let's look at this particular question on short tandem repeats here. So it says here, identify the sequence which is a short tandem repeat. And this is a typical multiple choice question. And if you look at them very, very carefully, you can see that the answer is ATTCG, there, ATTCG, ATTCG. So we can see here that the short tandem repeat here is a sequence of 5 bases. And in this case, it's repeated 3 times. So the answer for this one is D.